Bismillahirrahmanirrahim. Dear students, Assalamu alaikum. Today we are going to discuss glycogenolysis, that is the breakdown of glycogen molecule. In the previous lecture, we discussed the glycogenesis, which is the biosynthesis of this glycogen molecule from glucose. This occurs in the fed state, and this glycogenolysis occurs in the fasting state. This glycogen molecules results in the formation of glucose 1-phosphate and ultimately glucose in the liver and glucose 6-phosphate in muscles. We should be very clear that glycogenesis and glycogenolysis both are different pathways. They are not the reversal of each other, but the separate group of enzymes catalyze the glycogen biosynthesis and glycogen breakdown. This glycogen phosphorylase, which is a regulatory enzyme in glycogen breakdown, as glycogen synthase enzyme is a regulatory enzyme of glycogenesis. Both enzymes occurs in two forms, A and B form. Glycogen phosphorylase A is phosphorylated and glycogen synthase A is a dephosphorylated or non-phosphorylated form. The glycogen synthase A and glycogen phosphorylase A are the active form. So, the active form of glycogen phosphorylase is a phosphorylated and active form of glycogen synthase is dephosphorylated. As I have already told you, glycogenolysis is not the reverse of the glycogenesis but is a separate pathway. The glycogen phosphorylase catalyzes the rate limiting step of glycogenolysis, which is the phosphorylatic cleavage of alpha-1,4 linkage of glycogen to yield this glucose 1-phosphate. There are different isozymes of this glycogen phosphorylase which occurs in liver, muscles and brain. They are encoded by different genes. This glycogen phosphorylase requires pyridoxal 5-phosphate as its coenzyme. Pyridoxal 5'-phosphate is an active form of vitamin B6. In glycogen phosphorylase, it is the phosphate group that is catalytically active. The terminal glucosyl residue from the outermost chain of the glycogen molecules are removed sequentially until approximately 4 glucose residues remain on either side of alpha-1,6 branch point. Means there will be a sequential removal of terminal glucose from glycogen molecule till the 4 residues remain on both sides of the branch point. Then the debranching enzyme will act. This debranching enzyme has 2 catalytic sites in a single polypeptide chain. One is the glucan transferase that transfers a trisaccharide unit from one branch to the other and exposing the alpha-1,6 branch point, and other is the debranching enzyme, that is alpha-1,6 glycosidase, that catalyzes the hydrolysis of 1,6 glycosidic bond to liberate the free glucose. This can be better understood in the next slide. Here we have seen the sequential removal of glucose molecule from the outer chain of the glycogen molecule till on the both side we have 4 glucose residues. 1, 2, 3, 4. We know that this is the alpha 1 6 glycosidic link, and these linkages are alpha 1 4 glycosidic linkage. As we have already discussed, the terminal glucosyl residues from the outermost chains of the glycogen molecules are removed sequentially until approximately 4 glucose residues remain on either side of alpha 1 6. अब ये देखें, यहाँ पे भी 4 है, इधर भी 4 है. Now the debranching, which has 2 catalytic sites in a single polypeptide chain, that is glucan transferase and the alpha 1 glycosidase. This is the debranching enzyme. One, which is the glucan transferase, transfers a trisaccharide unit from one branch to the other, and this exposes the 1,6 branch point. I just repeat, trisaccharide transferred to the adjacent chain, and by this way this alpha 1 6 is exposed, and now this alpha 1 6 glycosidase act on it and causes the hydrolysis of alpha 1 6 glycosidic bond to liberate the free glucose. Then phosphorylase action can proceed, that is the sequential removal of glucose molecule from the outermost chain of the glycogen molecule. Again, we coming back to this. Now this glucose 1-phosphate will isomerized into glucose 6-phosphate by phosphoglucomutase, as it is a reversible reaction. Also remember that the combined action of this phosphorylase and this debranching enzyme lead to the complete breakdown of this glycogen molecule. This glucose 1-phosphate, as I have already told you, is converted to glucose 6-phosphate by phosphoglucomutase. Now this glucose 6-phosphate in exercising muscles or in the muscles enter the glycolysis and yield the energy. But in case of liver, this glucose phosphate convert into glucose by the action of this enzyme, which is glucose 6-phosphatase present in liver. This enzyme is not present in muscles. That's why in muscles glucose 6-phosphate is the end product, and glucose is not formed in muscle, as this glucose 6-phosphatase is not present in muscle, but as it is present in the liver, this glucose 6-phosphate will convert into glucose, and so it contribute to the blood glucose level. This glucose 6-phosphatase is in the lumen of smooth endoplasmic reticulum, and the genetic defect of glucose 6-phosphate transporter can cause a variant of type 1 glycogen storage disease we will discuss. This glycogen granules can also be engulfed by lysosome. Glycogen, where acid maltase catalyze the hydrolysis of glycogen to glucose, and this may be especially important in glucose homeostasis in neonates. Genetic lack of this lysosomal acid maltase causes type 2 glycogen storage disease, also known as Pompe's disease. Again, I am repeating, This glycogen molecules or the glycogen granules can also be engulfed by a lysosome, which contain acid maltase enzyme, which catalyze the hydrolysis of glycogen to the glucose, and this is very important in the glucose homeostasis in the neonates. Lysosomal catabolism of glycogen is under the hormonal control. Here we can see the formation and degradation of cyclic AMP. Remember, cyclic AMP integrate the regulation of glycogenolysis and the glycogenesis. This cyclic AMP is formed by the activation of this adenylyl cyclase, and this ATP convert into cyclic AMP, and this cyclic AMP is hydrolyzed to 5' AMP by phosphodiesterase, formed by the activation of adenylyl cyclase and degraded by the phosphodiesterase. This is the role of cyclic AMP. As we have already discussed, cyclic AMP integrate the regulation of glycogenolysis and glycogenesis. This cyclic AMP control the glycogen phosphorylase and glycogen synthase, which are the principal enzymes regulating the glycogen metabolism. These both enzymes are regulated in opposite direction by allosteric mechanism and by covalent modification. This covalent modification occurs by reversible phosphorylation and dephosphorylations of enzyme protein in response to hormone action. Phosphorylation of glycogen phosphorylase increase its activity. The activity of glycogen phosphorylase is increased as a result of its phosphorylation, and the activity of glycogen synthase decreases as a result of phosphorylation. Means its active form is dephosphorylated and its active form is phosphorylated. Both these enzymes occur in two forms, A and B and A and B. The active form of both enzyme is A form, which in case of glycogen phosphorylase is a phosphorylated form, and which is in the case of glycogen synthase is a non-phosphorylated form. This phosphorylation is increased in response to cyclic AMP. The detailed role of cyclic AMP, insulin, glucagon and epinephrine we will discuss in regulation of glycogenesis and glycogenolysis. Thank you very much.